Hi, my name is Dr. Adam Rich, and I'm a faculty member in the Department of Biology. I'm going to tell you about a course that I teach, a lab course called Systems Physiology for our undergraduates, and Advanced Physiology Lab for our master's level graduate students. In this course, students do research. There's only about a dozen students, and we do a bona fide, authentic research project that is meant to be modeled after a real research project that might happen in my lab or in someone else's lab. We begin the course with really a seminar, and then we develop a question. That question is really open-ended, and it's something that's never been done before. Then we divide into 3 research teams, each led by one person, typically a graduate student, and each team has its own individual objectives. Those objectives combined lead into the overall objective of the course. The course is really meant to model and to teach the process of doing science. So we have plenty of mistakes, lots of frustration, but everybody comes a long way in that they learn the process of science, they develop problem-solving skills and critical thinking skills. We really want to, in this course, push the idea of integrative problem-solving that I think is important for anything that comes in the future for these students, whether they pursue a research-based career or a clinical career or something else, something in between.
So basically right now we are washing off the primary antibody of first set of 5 DPF fish. And the whole process of histochemistry broken down is well, first we have to wash them, and then we have to put primary antibody on, and then wash that off, and put secondary antibody on that, and then that is what's going to show up. So the secondary antibody binds to the primary antibody, and the secondary antibody is the one that fluoresces. So then if everything works out good, we should be able to see what the whole GI tract of these 5 DPF fish will look like, and then later on what the 7 DPF fishes look like.
Technology is really important in modern research. In this lab, we have to actually see how the organs function. So we use microscopes and we use digital imaging, and then we analyze movement in the images with computers. Technology is also really important for teaching, so that when we talk about science, if we can actually see what's going on, that's fantastic. So we have these microscopes with cameras, and the cameras are hooked up to larger screen monitors so that the person looking through the microscope can see what's going on. But then the other team members and the other scientists in the room can actually talk about it. So that's one example of technology. And we have many others all around the lab. And every group is using really modern technology to implement these modern techniques to look at gene function using the zebrafish model.